from United Christian College, Column East. And today we would like to introduce you our project about a synthetic biology approach for cancer screening. Before we start our presentation, I would like to ask how many of you have your close family members or close friends suffering from cancer? May you put up your hands? Okay, thank you. We can see that actually cancer is a common disease. We can actually see that cancer is a common disease among modern people. I believe not only the one who is suffering from cancer know how hard it is to face this difficulty, but also it's very tough for those people around him or her to face this disease with them together. As we know that cancer is the first leading cause of death in the world, which countless people are now suffering from. According to the data provided by the Global Can Estimate, 19.3 million of new cancer cases are found in 2030, and 10 million of these cancer patients lost their life due to different kinds of cancer, especially for lung, breast, and colorectal cancer. If this situation persists, the NIH National Cancer Institute believes that the number of new cancer cases per year will rise substantially to 29.5 million in 2040. Moreover, 16.4 million people will die due to cancer per year at that time. It is no doubt that we need to find a way to put our hands to this problem. You may ask, are still current solutions such as biopsy or screening enough to deal with cancer detection? Although those detection methods did help many people to find out their health problem, we realized that numerous recent methods are not too high in accuracy or user-friendly enough. Such as biopsy needs a close-up surgery which is invasive, patient may feel uncomfortable in the process. Also, screening may have a higher chance of false positive or negative, which means it's not high in accuracy. And for post-symptoms detection, you may have missed the critical time for medication treatment already at the time. As a result, we can see that it's important to provide a more user-friendly and higher accuracy detection method to the public. So, our project aims to make an accurate and non-invasive cancer screening kit, which not only providing reliable results, the patient may also feel more comfortable during the process. Secondly, we aim to detect cancer in an early stage in order to prevent patients' condition getting deteriorates. This can lighten the burdens of healthcare systems and also decrease mortality rates. And now I will pass the time to other members to talk about how we are going to achieve these aims.
So first of all, we have designed a GM E. coli that detects cancer antigens and give out signals. When there is no cancer antigens detected, the bacteria are resting state and only proliferates at a lower than normal rate. Because the cell is a double knockout of two important genes. On the cell surface, we have MRP proteins to detect cancer antigen. MRP stands for modular receptor platform. If the MRP detects the cancer antigen with its antibody detection domain, it will activate a promoter on its DNA. As a positive result, GFP, green fluorescence protein, will be expressed, and the two knockout genes will be rescued in order to restore normal cell proliferation. This will also give off visible signals. So let me introduce the MRP, the modular receptor platform. The protein is designed according to a study published in 2018. The MRP protein is expressed to locate on the E. coli's membrane. Each MRP monomer has an extracellular single domain antibody and a split Cat-C DVD. The platform is flexible in detecting different cancer antigens and even other diseases because of the antibody engineering technology. When antibody binds an antigen, the DVD will dimerize to bind the Cat-C operator. The Cat-BA promoter is activated to express downstream genes, including GFP and the other proteins. So, other than GFP, the Cat-BA promoter also controls cell proliferation too. The GM bacteria, as we mentioned earlier, has its own ICLR and ARCA genes knocked out in its genome and grow slower than normal. When the MRP activates the CATBA promoter, the knockout genes are expressed. This is meant to rescue the GM bacteria from the suppressed growth to a normal self-growth rate, so that more E. coli is presented. In a shorter time, therefore, the visual signal provided by the GFP is amplified and it makes the result more observable. Other reason why we need to restore the normal cell proliferation is because two positive signals together can increase the reliability of our kits. So in conclusion, detection of cancer antigen at the MRP will induce two responses, the GFP expression and the self-growth rescue. The GFP expression provides visual signals by emitting green-colored light. The second response is to express ICLR and ARCA to restore normal self-growth so it can amplify the visual signal. So here is our plasmid insert for our GM bacteria. The MRP is always expressed and the expression of GFP, RCA, and ICLR are under control by the CATBA promoter. Thank you.
So the experiments we're going to do are divided into two major parts. So first of all is the MRP expression, and secondly is the cell proliferation. We will transform the MRP plasmid into BL21, which is an expression strain E. coli. Then we would like to confirm whether the plasmid is in the cell. To achieve this, we would check clone by cloning PCR after the transformation. Unfortunately, the transformation we did yesterday failed. Once we confirm that the plasmid is successfully transformed, we plan to induce protein expression by IPTG via R0010 lac operon promoter, which is upstream of this experimental construct. Here, the lac operon promoter is circled in red. Since the expression of MRP would not be visible, we added a reporter protein downstream MRP. The red fluorescent protein is under the control of the same promoter. We expect that when the RFP is expressed, that is when the culture turns red, our target protein MRP is also expressed. But of course, we hope the expression can be confirmed by SDS page. In SDS page, we hope to confirm the expression of the MRP protein by running SDS page gel. However, as secondary school students, we do not have the access to SDS page equipment or reagents. So we would like to ask for kind help here as a form of collaboration.
For the cell proliferation part, we ordered the Kyo collection from the Japan Institute for our experiments. The Kyo collection are systematic single-gene knockout mutants of E. coli K12. They are ARCA, ARCA knockout, and ICLR knockout respectively. To test if the KO can be rescued from the suppressed growth rate, we will transform a plasmid carrying the knockout gene to stimulate the active state. There, we will check if the plasmids are successfully transformed into the KO E. coli by cloning PCR and gel electrophoresis. The second experiment we'll do is a self-growth assay to see if knockout and rescue have any effect on the strains. We will culture wild-type E. coli, ARCA knockout E. coli, ICLR knockout E. coli, and rescue ICLR knockout E. coli and measure their optical density over time by plate readers. The growth curves will then be generated. by the growth curves obtained from the self-growth assay, we will start the first step of our modeling. We will input the growth curves of the 4 KO strains to growth model, which is an R package for fitting growth curves of microorganisms. Then, the model will generate the growth parameters of 4 KO strains, which are lag time, maximum growth rate, and maximum cell density. We designed that our GM bacteria is an ARCA ICLR knocked out E. coli because this double knocked out is both non-lethal and has a lower growth rate according to a reference study. However, the KIO collection has only single gene knockouts, so we cannot knock out on our own. To make up for that, we would like to estimate the growth parameters of double knockout strain by putting the growth parameters of the 2 single knockout strains into a model. However, we do not know how to stimulate the growth parameters of the double knockout E. coli, so we would like to collaborate with any teams that are able to offer us help. Although we are restricted by our knowledge and the access to some of the methods, we believe in the strong iGEM community. So, we would like to modestly ask for your help. If you have any suggestions, we are very, very thankful to hear them. At last, we would like to solemnly thank you for listening to our presentation and any further collaboration.
Guys, do you have any questions for this game?
Yes.
I want to ask, since like different cancer cells have different markers, how can your construct detect different types of cancer cells?
Because we can, in the MRP protein, we can change, by changing the antibody of the MRP protein, we can detect different antigens of different cancers.
But if you already don't know which type of cancer you have, how do you know which? If you already don't know which cancer you have, how do you know which antibody to put in to detect that sort of cancer?
Maybe we will try and ask some patient who is recover from cancer to use this case. Maybe the patient at the beginning, he or she has colorectal cancer, and we may, after he or she recover, and we may also ask he or she to use our kit to check whether he or she will has the cancer again. So we will use specific, antibody.
You mentioned that knocking out both of the genes doesn't necessarily arrest the cell cycle. Like it doesn't stop the cell from proliferating, right? But then how slow does it get, and whether that's sufficient of a difference between normal growth rate and arrest rate? Can you differentiate them? Is the arrest strong?
In the research paper that we read, the growth rate of double knocked out E. coli will be reduced by 38%. And we'll do the self-growth assay again so we can test off the growth
trying to challenge with anything. I just wanted to ask, do you guys have any thoughts, or have you had some actions on the human practice side? No, if you guys don't, it's okay. Yeah, we're just curious, pure curiosity. If you guys don't, it's okay.
We have planned to interview some professionals of in the medicine field, and we have also have interviewed one of synthetic biologists about our project, and he does give us some advice, but we didn't add it.
Sure.
As you have mentioned, the protein transformation is failed yesterday. So my suggestion is you better think of the backup plan, because otherwise it is the main component of your project, and if that fails, and you cannot continue to do
It would be very
the plan.
difficult.
Yes.
Yeah.
Do you guys have any more questions? Do you have a question?
They're not questions. It's just some comments that I think the judges will ask you. First of all, because your project is trying to develop a non-invasive detection assay, as you mentioned, and I wonder how you intend to achieve that. Because if you want to get the cancer antigen, if there is any, that's my second question actually. If you want to get the cancer antigen from the cancer patient, you need to use some invasive, although maybe minimally invasive, like methods to get the cancer antigen, maybe from the blood or other bodily fluid. Yeah, so that's one, I think, very general question that the judges might, like, I think, must ask you. And then the second question, actually relating to the question that was asked by the EST team. You the reason why cancer is so difficult for us to compare against is because our immune system is not that good at recognizing them and therefore generating antigen against cancer. So that's why maybe that's one of the reasons why the question there asked which type of cancer that you're trying to detect. And you have to show evidence to the judges, like, oh, we are trying to, like, we know there is antigen, cancer antigen that can be detected. I mean, of this cancer type that can be detected by that can be generated by our body, and therefore we can detect it. Yeah, so I think these are some questions that you have to consider.
All right, thank you very much, UCCKE.